Good afternoon. I would like to thank Director Cruz for her kind invite to me to speak this afternoon. I'm very honored. I will be talking about mechanistic studies, clinical trials, and the future of both. First, I will advocate strongly for basic research, especially among junior investigators. I will outline one career that progresses from basic research to conducting a clinical trial. I will then show the potential importance of phase 2 clinical trials, which may or may not be futility trials, as a lead to phase 3 efficacy clinical trials. Finally, I will present a phase 3 clinical trial, which has been designed to make sure we obtain important data, even if we do not detect differences between our two treatment arms. In 2014, NIH began a major effort to enhance the quality, relevance, feasibility, and transparency of NIH-funded clinical trials. The definition is a research study in which one or more human participants are prospectively assigned to one or more interventions, which may include placebo or other control, to evaluate the effects of those interventions on health-related, biomedical, or behavioral outcomes. This definition and the fact that many people in rehabilitation want to do intervention studies has led to an increase in the number of studies classified as clinical trials. I personally am not yet convinced that this very expansive definition of a clinical trial is helpful. For example, a single-arm intervention study with a health-related biomedical or behavioral outcome is a clinical trial by this definition. I think there is still an important place for mechanistic science in which the focus is understanding mechanism. Equally important, understanding mechanism may well inform clinical practice. Clinical trials involve significant bureaucratic overhead and should be approached with caution, in my view, early on in the scientific career. Here, I'm going to outline the career trajectory of Dr. Jason Kutch, who is at USC. In 2011, he started as a tenure-track assistant professor in biokinesiology and physical therapy at UIC. You can see on the top left part, in 2014, he published initial studies from the MAP study of chronic pelvic pain, looking at the network showing altered SMA resting activity in pelvic pain patients. In 2015, he published two important articles in the Journal of Neuroscience confirming SMA role in pelvic muscle control. In the top slide, which is the top caption for the slide taken from his grant, he submitted a grant, and in 2017, this was funded. It was to look at how resting SMA function impacts active control of muscles on the pelvic floor for pelvic pain. It's great news. In 2019, he was awarded tenure at USC, and just recently, he's been awarded a clinical trial, which is an intervention study in women to reduce pelvic pain by stimulating over the SMA area using RTMS. So you can see over a decade, Jason has gone from very elegant basic research to now implementing a clinical trial. I now want to move on to look at Sparx II, which is a phase 2 clinical trial in Parkinson's disease. The purpose is to study the feasibility and safety of high-intensity endurance exercise. The question we posed is, can patients who have not yet taken medication for Parkinson's disease exercise safely at high intensity, 80 to 85% of maximum heart rate, for 4 days a week? Also, at this time, the effect on the motor signs of Parkinson's disease had not yet been established. Here we have the consort diagram for Sparx II. I want you to notice that we had a high-intensity condition, 80 to 85% of maximum heart rate, a moderate intensity condition, 60 to 65% of maximum heart rate, and then the third treatment arm was usual care, where people were just asked to carry on doing what they had been doing previously. In this slide, we show that people with Parkinson's disease can exercise at 80 to 85% of maximal heart rate. And you'll notice the error bars are very small. And they can also exercise at 60 to 65% of maximum heart rate. So we had shown the feasibility of being able to exercise at the assigned heart rate. Our target number of days was 4. You can see here that the 60 to 65% were just above 3.5 days a week. There was no statistically significant difference between the 60 to 65 and the 80 to 85% group. So we showed that people can exercise for the required number of days. And then thirdly, we looked at the Unified Parkinson's Disease Rating Scale. This is an assessment of how Parkinsonian a person is. And here we're plotting the 6-month change in UPDRS motor score. At 6 months, there was virtually no change in how Parkinsonian the people were for the 80 to 85% group. 60 to 65% the group had progressed further with their disease. The usual care group had progressed even further. A low score is good. We then did a futility analysis, and it was shown that the difference between the maximum heart rate, the 80 to 85% maximum heart rate and usual care was not futile. The 2.9 point difference that was deemed not futile. The difference between 60 to 65% was deemed futile. So this gave us the rationale for moving on to a phase 3 clinical trial. Now, to write a phase 3 clinical trial, certainly with NINDS, you need a phase 2 successful clinical trial. You also need to outline the mechanistic underpinning of the science to inform the phase 3 clinical trial. Dr. Michael Schwarzschild sent me a very nice email the other day, and it really captures what one needs to have to set up a phase 3 trial. And it says aerobic exercise is the treatment with the most compelling evidence of laboratory, which I will show you, risk factor, which I will show you, and human trial evidence, which was the phase 2 clinical trial I've just discussed. So if we look at the slide here, it shows that monkeys with the highest levels of physical activity had the least damage to nigrostriatal dopaminergic neurons. So here we have a PET scan, and these are monkeys who were doing low levels of activity, and you can see that on the left side here, there is very, very little dopaminergic uptake in striatum. The intermediate activity, you have some, and then the monkeys doing the high activity had the closest to a normal brain, and you can see dopamine uptake in both caudate and putamen. Dr. Schwarzschild wrote a very important paper back in 2005 on physical activity and the risk of Parkinson's disease. And in the conclusion, the study suggests that either higher levels of physical activity may lower the risk of Parkinson's disease in men. It may be the case that men predisposed to PD tend to avoid strenuous physical activity, but many studies have come out since then showing that high levels of physical activity tend to be linked to lower levels of risk. So given we had all this information, we were ready to propose Sparx III, and we were successful. And so the study objective is to establish the efficacy of high-intensity endurance exercise as a first-line therapy for recently diagnosed people with Parkinson's disease. The key question is whether there is an additional benefit of exercising at high intensity in terms of slowing the progression of the signs of the disease beyond the well-documented benefit of treadmill training on general parameters of fitness, gait, and functional mobility. Here is the original design for Sparx III, the design in the grant. We have physician referral, screening and scent, initial baseline assessment, stratified randomization, and then we had 2 treatment arms, 80 to 85% and 60 to 65%. Getting the control group was the hardest part of the design. We discussed it for perhaps at least 2 years before we decided to use the 60 to 65%. The primary outcome was at 6-month master assessment, and we chose this based on the Sparx II data We really would probably have preferred to use the 12-month master assessment, but we decided to write the grant based on pilot data to make sure that the reviewers knew that we had pilot data for the outcome at 6 months. So the grant went to council, and council is acutely aware, NIH is acutely aware, that phase 3 studies often don't work. So here we have a summary of phase 3 studies that failed to replicate phase 2 findings. These are drug studies, but I think one can learn from them. And you can see there are 7 major studies here, and they all failed, even though they had a phase 2. This is really quite sobering. PD clinical trials on neuroprotection in the 20th century. Also, this is very, very bleak prospects in a sense. So you can see, if you look at the results at the bottom, as we go across, it's either futile, may warrant further study, did not slow progression, unlikely to modify progression, did not slow progression, stopped early. We look at clinical trials in stroke. There was the EXCITE study in 2006, which was very successful. But if we look down across the studies from Everest, GRASP, LEAPS, iCare, EVERT, ATTEND, REM, RATTLES, INTENSE, you can see most have no significant between group difference. I was privileged enough to have extensive dialogue with Kathy Lang, Steve Wolf, and Carolee Winstein in preparing to present this talk. And they agreed that this is a reasonably fair summary of the state of the field. You should all know there are 2 excellent review papers that draw similar conclusions, which I've listed below. So council, my program officers intervened, and we had lengthy discussion on how to modify the design of Sparx II. The concern, are you adequately statistically powered? 6 months was a concern to us, it was a concern to the statisticians, and it was a concern to NDS. This is the MDS UPDRS that I'm showing on the left. The differences are a little bit bigger than the UPDRS, which we used in Sparx II, but at 6 months, the difference between the 80 to 85 and the 60 to 65 was certainly questionable. So the solution, they suggested changing the primary outcome variable to 12 months. They also added an extra 6 months of supervised exercise. So the study now is a 2-year intervention study, which we could not have proposed in a 5-year grant. So Dr. Patterson and I realized some of these issues, but 5 years sounds a long time, but to fully implement a large intervention study is hard. Another concern, perhaps concern is the wrong word, but another interest is what happens when you remove all of the interventional support. So when you're part of a phase 3 clinical study, you're motivated to take part. Now, as all of you in the audience full well know, exercising is hard. So here are at least 10 excuses that I got from the Heart Foundation. Wonderful website because they provide answers to all of these excuses. So for those of you who struggle to exercise, I suggest you go to the Heart Foundation website, and whatever your excuses, they have an answer. So what we decided was to add an extra 6 months of unsupervised exercise to look at attrition. So what this will allow us to do is to determine whether people actually continue to exercise. So here we have the updated design, the one that we're currently doing. So we have baseline assessment, 6-month assessment. Our single primary outcome is at 12 months. So minimally clinically important difference is very important in clinical trials. And I think as we move to the future, we need to do much more work on minimally clinically important difference. In Sparx III, it's 3.5 points on the motor section of the MDS UPDRS. And then we're going to be assessing at 18 months. And then this is the observational period. This is the progression in the MDS, which we predict for the group exercising at 80 to 85%. And this is the prediction for the group exercising at 60 to 65%. The PPMI number here of 6.3, this is an observational cohort of people with Parkinson's disease, and after a year, they actually progressed 6.3 points. Now, I want to talk about the superiority hypothesis at length. So to fail or not to fail, that is the question. If Sparx II fails to detect the difference between the two groups on the MDS-UPDRS Part 3 at 12 months, is it a failed clinical trial? Hopefully the chat will start to light up now, because I'm sure everybody has opinions. And if none of you do, I know for certain Catherine Lang does. Now, editors of top journals will probably say yes. Many statisticians will say yes. Multiple time outcomes and multiple outcome measures can lead to erroneous conclusions by chance. This makes statisticians nervous. It's why when you have two primary outcome measures, you are required to divide alpha by 2, which then makes it harder to get significance. But these are very legitimate and very important concerns. In my view, the rehabilitation community needs to take a principled position on how to interpret large clinical trials if the superior hypothesis turns out not to be supported. In Sparx II, we're placing a lot of emphasis on secondary outcomes, modifying variables, and mediating variables. So we have a biomarker that may work. This is the DAT scan. Again, here you have caudate and putamen activated bilaterally in a healthy person. Here is a person with Parkinson's disease. The expectation is that the DAT scan will decline less. The dopamine uptake will decline less in people exercising at 80 to 85%. We have a whole panel of inflammatory and neurotropic biomarkers that may prove to be sensitive, and there are major initiatives to develop objective sensitive biomarkers from blood. We have also added a set of sensitive functional outcome measures that may well be the most sensitive measures of all. This is driven by the pioneering work of Drs. Horak and Mancini, who are developing very important measures of turning time, arm swing, and other measures. We will be collecting genetic data on all the participants. I'm very interested, and I think it's very important that we consider responders versus non-responders. Studies can fail to meet the primary efficacy outcome. In a very real sense, they have failed at the population. So what conclusion can we draw? In Sparx II, we will have extensive biomarker data to possibly tease apart why some patients respond better than others. So if you look at this slide again, you'll see in the group exercising at 80 to 85%, 3 people did really, really well. Not only did their disease not progress, it actually was less. Others progressed more. And it's this differential response which causes difficulties for phase 3 clinical trials because of the variance that is introduced by it. And I think we need to understand responders versus non-responders better. I'm going to go off on a slight tangent now, just make one other point. And that is this issue becomes much harder in some areas of treatment. So suppose you are given an implantable brain device that improves your function dramatically. Suppose you are in a clinical trial that fails to meet its primary outcome variable, and you are told the device is no longer supported, and the neurosurgeon needs now to remove the device. What next? How will you feel? I think as we look forward in the area of rehab over the next 30 years, as we envision a better functional future, the role of ethicists will become much more important. Also want to talk about interim power analysis. Normally, interim power analyses are performed to see if there is sufficient reason to carry on the study. If an interim power analysis shows that the study will not meet its goal, it may well be stopped. This happened to the last phase 3 clinical trial on neuroprotection. In Sparx II, we have proposed an interim futility analysis that if the mean difference we observed is consistent with our hypothesis, but we have increased variability, then we will increase our N. The logic here is that Sparx II was based on 3 sites. Sparx III is based on 29 sites. And as you expand the number of sites, with more people doing tests and delivering the intervention, you may inflate your variability more than you thought. And we'll be able to look at this. The idea is if we're correct about the expected mean differences, but are wrong about variances because of how sites implement the protocol, then we will increase the sample size. I want to talk about within treatment arm analysis, which I think is something that should be developed. Suppose both the control arm and the treatment arm work. Suppose the treatment arm clearly exceeds the MCID of 3.5 points, which is what we've set in Sparx II. Suppose it's 4 points. Suppose the control arm does not exceed the MCID, 2 points, for example. How should one interpret these data? So at the end of the day, there's no difference between the groups, but one clearly exceeds the MCID, one doesn't. So in terms of summary and recommendations, I would like to advocate strongly for basic research, especially among junior investigators. There is nothing so practical or functional as a good theory. We need mechanistic biomarkers. Continue to push the rigor and transparency that the methods of clinical trials bring. However, discuss removing the clinical trials framework from studies not designed to change clinical practice. Develop the pathway from phase 2 trials, which may or may not be futility trials, to phase 3 efficacy trials. There are still huge problems here and much work to do. Make sure that clinical trials can be informative, even if the superiority hypothesis fails, since this is a distinct possibility that it can fail. Develop adaptive statistical models to look at interim findings of large clinical trials, both in terms of shutting them down and also providing additional support. Develop consistency across NIH institutes with respect to length of time for grant support. 5 years is often too short, and some institutes have recognized this explicitly with grants that go beyond 5 years. I want to thank NIH, NINDS for all the support. Without them, clearly, none of this work could be done. I've also received a lot of support from Northwestern and the Dean. I want to thank you very much for your support, for your time. Thank you.